Good afternoon, students. Today we are going to start the next topic, that is transcription and translation. So first we are going to deal with the topic that is transcription, right? Now when I talk about transcription, transcription means transcribe, and the meaning of transcribe is copying. So we all know that DNA is a genetic material, correct? This DNA generally contains genetic information. Now there are three types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA will copy the genetic information from the DNA and will it pass to the ribosome for the protein synthesis, right? So the process where the messenger RNA copies the genetic information from the DNA and passes to the ribosome for the process of the protein synthesis is generally called transcription, right? So transcribing is copying. You can also say that there is a formation of a mRNA on the DNA template strand. Template is the old strand of a DNA. So the synthesis of the messenger RNA on the old strand or the template strand of the DNA is generally called transcription. Okay, so let's talk about transcription in detail. When I talk about this transcription, transcription requires many different components. I have mentioned here, right? So transcription requires following important component. First is promoter and terminator. Now what do you mean by promoter? Promoter is a small sequence of a DNA where RNA polymerase enzyme comes and binds, right? So a small sequence of a DNA where the RNA polymerase enzyme comes and binds is called as promoter, and promoter is always oriented at the phi dash end. Second, terminator. Terminator is a sequence of a DNA where the transcription process stops, right? So terminator is a small sequence of a DNA where the transcription or the protein synthesis process stops. It is oriented at the 3' end. Okay. Next one is nothing but your two different strands of a DNA. One is called antisense strand, or second is generally called as coding strand, right? Antisense strand is called as template strand and coding strand is generally called as sense strand. Now what is antisense strand? The strand of a DNA which plays an important role in the RNA synthesis is generally called antisense strand or the template strand, and it is oriented from 3' to 5' direction, correct? Second one is generally called as sense or the coding strand. The strand of a DNA which does not expresses or does not help in the synthesis of the RNA molecule is generally called coding strand or the sense strand, and it is oriented from 5' to 3' direction. Right, so two strand, one is called antisense strand or the template strand, and second is called as coding strand or the sense strand. Right, now when you talk about the eukaryotic cell generally has two sequences or split it up into exon and intron molecule. Now when I talk about exon and intron, what are they? Right, the DNA sequences which, or you can say the DNA sequences or the molecule, the DNA sequences which expresses, right, or appears in a mature RNA molecule is generally called as exon, whereas the DNA sequences which are not expressed or which are not appeared in a processed RNA molecule is generally called as the intron, right? So I have 6 different components here lined up. One is called as promoter, second is terminator, next is antisense or template strand, coding or sense strand, exon molecule, and intron molecule. These are the basics of transcription. I mean to say, to start the transcription process, we should know the basics of all this component. Now let's start about the transcription, right? So in transcription, there are 3 important steps, right? First is called as initiation, second is called as elongation, and third is termination. Now, first, initiation. Initiation is generally done, or initiation is generally brought about by a following process. The name of the enzyme is RNA polymerase. RNA polymerase is a kind of an enzyme which will come and bind at the phi dash end, which will come and attach to the phi dash end or the promoter site. Right, so you know very well RNA polymerase is the enzyme, correct? So initiation is brought about by RNA polymerase enzyme. RNA polymerase enzyme will come and bind at the phi dash end or the promoter site and will begin the process of the transcription. I mean to say protein synthesis starts. Second step is generally called elongation. Once the RNA polymerase enzyme comes and binds to the promoter site, now the two strands of a DNA generally get separated from each other, right? The double-stranded DNA or the two strands of the DNA generally start separating from each other. Now, according to the base sequences present on the template strand, the original old DNA strand is called as template strand. So, according to the base sequences, base pairs present on the according to the base sequences or the base pair which are generally present on the template strand, new complementary RNA nucleotides are selected and joined one after other to form a long messenger RNA molecule, right? Okay, so what happens in the second step? Second step is called elongation. The promoter, the RNA polymerase enzyme will come and bind to the promoter at the 5' end. Now the double-stranded DNA will separate from each other. Now, according to the base pair present on the template strand of a DNA, new RNA nucleotides are generally selected and joined one after the other to form a messenger RNA molecule. This is generally called elongation. Now, third step is termination. Now, what happens in the termination is that the RNA polymerase enzyme, this is the RNA polymerase enzyme. The RNA polymerase enzyme will start, right? The RNA polymerase enzyme will start moving from your 5' end to the 3' end. And when this RNA polymerase enzyme comes at the terminator site or the 3' end, the newly constructed RNA falls off and the enzyme RNA polymerase also falls off and the transcription stops, right? So termination is a type of a process where the RNA polymerase enzyme will start moving from the 5' to 3' end. When it reaches to the 3' end or the terminator site, the newly constructed RNA molecule and the RNA polymerase enzyme falls off and the transcription generally stops. That is, protein synthesis generally stops. These are the three steps where the transcription takes place: initiation, elongation, termination. In initiation, RNA polymerase will come and bind at the 5' end and will start the protein synthesis. In elongation, double-stranded DNA separates. According to the base pair of the DNA template, new RNA nucleotides are selected and joined one after the other to form a long RNA molecule, that is mRNA molecule. And in third step, when the RNA polymerase reaches at the 3' end, the newly constructed RNA molecule and the RNA polymerase enzyme falls off and the transcription stops. Now, when you talk about the prokaryotic cell, right, isn't it? In a prokaryotic cell, we have seen that is, in prokaryotic cell, you have a 70S type of a ribosome, and eukaryotic cell, you have a 80S type of a ribosome. So, in prokaryotic cell, the matured RNA which is generally formed, that is the messenger RNA which is generally formed, does not require much processing. But as we know that in eukaryotic cell, that is the advanced cell, once the messenger RNA molecule which is a non-functional RNA is formed, further processing is required. So in eukaryotes, you can see it undergoes three important types of processes, right? First is called as splicing, second is called capping, third is generally called tailing. Now, what is splicing? The removal of the introns from the RNA, or right, from the removal of the introns from the DNA, you can say, is generally called as splicing, right? Okay, so I told you what are introns and what are exons. These are the exons and these are intron. Exon, intron, exon, intron, exon, intron, exon, right? So the removal of the intron is generally called as the splicing. Second step is generally called as capping, right? Now, addition of unusual nucleotide, that is methylguanosine triphosphate. There is the addition of unusual nucleotide. The name of it is methylguanosine triphosphate. That addition takes place at the 5' end. This is your 5' end, correct? So at this 5' end, there is the addition of unusual nucleotide guanosine triphosphate, and that process is generally called as capping, right? So capping is a process where there is the addition of unusual nucleotide methylguanosine triphosphate at 5' end. And last step is called tailing. The addition of adenylate residue at the 3' end. The addition of adenylate residue at the 3' end is generally called your tailing. So three processes takes place in your what? eukaryotic cell. One is splicing, that is removal of the intron. Second is capping, addition of unusual nucleotide methyl guanosine triphosphate at the 5' end, and tailing, addition of adenylate residue at the 3' end is generally called tailing. So here the transcription process takes place in such a way in a eukaryotic cell. Thank you. Next topic is translation. So as we discussed about transcription, we are going to discuss about the same translation. Now translation is transferring, correct? Now what is translation? The process where the sequence of the codons on the messenger RNA are read or they are being used and the amino acids so formed are linked with each other to form a long polypeptide chain is generally called as translation, right? So you know very well the sequences of the codon present on the messenger RNA, right? They will be read, they will be utilized, and similarly the amino acids will be formed and those amino acids will link with each other to form a long polypeptide chain. That process is called translation. Now when you talk about the translation, for the translation, first two important processes are first activation of amino acid, correct, and formation of amino acid transfer RNA complex, right? Second one is formation of a polypeptide chain. So let's talk about the first process, that is activation of amino acid and formation of transfer RNA complex. With the help of the enzyme amino acyl transfer RNA synthetase. With the help of the enzyme amino acyl transfer RNA synthetase, right, the amino acid generally goes and gets binded to the transfer RNA molecule at the 3' end or CCA end to form transfer RNA amino acid transfer RNA complex. So what is the name of the enzyme? Amino acyl transfer RNA synthetase, correct? So with the help of this enzyme amino acyl transfer RNA synthetase, amino acids are generally transferred or amino acids are generally attached to the 3' end or CCA end of the transfer RNA molecule to form amino acid transfer RNA complex. Second, formation of a polypeptide chain. For the formation of the polypeptide chain, you required 30S, correct? 30S kind of a ribosome and 50S subunit of ribosome. 30S and 50S type of a ribosome is required in a prokaryotic cell. Okay, you require amino acid transfer RNA complex and you also require ATP and GTP as a source of energy. Okay, so now we will start the 3 steps of translation. Similar step, that is first initiation, second elongation, third termination. So first step, initiation. The initiation step is generally brought about by the binding of RNA molecule on a small 30 subunit of ribosome. So what happens here? The ribosome, right? You have ribosome 30S and 50S. So initiation takes place by binding of the binding of RNA, right? Binding of a RNA on the small subunit of 30 subunit of ribosome, right? The initiation codon present at the 5' end is AUG. So the amino acid transfer RNA complex which has been formed, correct, will go and bind to the initiation codon AUG. Now this initiation codon AUG is specified by the UAC anticodon of transfer RNA. So we all know that codon is present on the messenger RNA and anticodon is present on the transfer RNA. Okay, now what happens is that once the amino acid transfer RNA complex goes and binds to what? Goes and, correct now, amino acid transfer RNA complex will generally go and bind to the AUG codon of messenger RNA, and this will be specified by the USG anticodon of the transfer RNA. Now the smaller subunit 30S, the larger subunit 50S will join with each other to form a 70S type of a ribosome. Now in prokaryote, that is 70S type of ribosome, the ribosome has mainly 3 important sites, very important. That is first amino acyl A site, second peptidyl P site, and third is exit site. Now, the amino acid transfer RNA molecule which has been formed will directly go and bind to the peptidyl P site, whereas the other incoming transfer RNA complexes will first go and bind to the A amino acid site and are further shifted to the peptidyl on the P site. So the first amino acid, right? The first amino acid, that is amino acid 1, transfer RNA complex. Transfer RNA complex. This will go and bind to the peptidyl site. Whereas the second amino acid 2, transfer RNA complex, will further go and bind to the amino acid A site and then shifted to the P site, that is peptidyl site. Now, the polypeptide chain which is generally released is always released from the P side. This is generally called first step, that is initiation. Correct? RNA molecule will go and bind to the small 30S subunit of your ribosome. Correct? Now, what will happen? The initiation codon is AUG. Amino acid 1 transfer RNA complex will go and bind to the AUG. Codon of messenger RNA will be specified by the UAC anticodon of transfer RNA. 30 subunit smaller, 50 subunit larger will unite with each other to form 70S type of ribosome. Ribosome has 3 sites, that is A site, amino acid, peptidyl P site, and exit E site. Okay. Now, second step is called elongation. The elongation is generally brought about by the formation of peptide bond or the peptide linkage between the two successive amino acids, right? And this is under the control or under the favor of an enzyme, peptidyl transferase enzyme. So peptidyl transferase enzyme will help in the formation of a polypeptide chain, that is formation of a peptide bond or a peptide linkage in between the successive amino acid. Due to the complementary nature of your codon and anticodon, amino acids are placed at the proper position. Now, what happens in the elongation is that the ribosome, so example, this is ribosome, right? And this is your RNA. So ribosome, the ribosome will start moving from the start codon to the stop codon. Start codon is initiation codon, stop codon is termination codon, 5' and 3'. So the movement of the ribosome from the start codon to the stop codon of the messenger RNA is called as translocation. That is called elongation. Third step is called termination. Now, what will happen when your when your ribosome reaches to the terminator site, that is at the 3' and the termination occurs, right? Now, to identify the termination codon in the releasing polypeptide chain, we require R1, R2, and S as the releasing factor. So, R1, R2 and S are the releasing factor which are required in identifying the termination codon and also in releasing the peptide chain, right? Okay, now once the termination takes place, the 30S smaller subunit and the 50S larger subunit separate from each other. This is your 30S and this is your 50S. This is smaller subunit, this is larger subunit. Now 30S will get separated from your what? It will get separated from the larger subunit 50S. This is called termination taking place in translation. So translation also requires 3 steps: initiation, elongation, and termination, right? Termination will only take place when the ribosome, that is 30S ribosome and 30S subunit and 50S subunit of ribosome will split with each other. They will get separate from each other and the translation will take place. Okay, thank you.